Imagine Earth billions of years ago. This was a time before plants and animals, a time when our planet was a very different place. The air was thick with gases like methane and ammonia. Volcanoes erupted, spewing lava and ash. This was the chaotic and volatile world where life first emerged. In this primordial soup, organic molecules like amino acids began to form. These molecules, the building blocks of life, came together in the vast oceans. They interacted and combined, eventually forming more complex structures. Clay minerals, abundant in this early environment, played a crucial role. They acted as catalysts, speeding up the reactions that led to the formation of primitive cells, the precursors to all life on Earth. These early cells, called prokaryotes, were simple and single-celled. They lacked a nucleus and other complex structures found in modern cells. However, they possessed the remarkable ability to replicate and pass on their genetic information. Over time, these early life forms diversified and evolved, adapting to the harsh conditions of the early Earth. Among these ancient prokaryotes were the ancestors of mitochondria. These bacteria-like organisms had evolved a remarkable ability. They could harness energy from their surroundings through a process called cellular respiration. This process, still used by nearly all life on Earth today, allowed these ancient bacteria to thrive in a world slowly becoming infused with oxygen. The story of mitochondria takes a dramatic turn with the theory of endosymbiosis. This theory, now widely accepted, proposes that a larger prokaryote engulfed a smaller energy-producing prokaryote, similar to the bacteria that would eventually become mitochondria. Instead of being digested, the engulfed prokaryote survived. This chance encounter proved mutually beneficial. The larger cell gained a reliable source of energy from its newly acquired resident. The smaller prokaryote, in turn, received a safe haven and a steady supply of nutrients from its host. This partnership, a remarkable example of symbiosis, marked a turning point in the history of life on Earth. Over millions of years, this symbiotic relationship deepened. The engulfed prokaryote, no longer independent, gradually transferred some of its genetic material to the host cell's DNA. This integration cemented their partnership, transforming the once free-living prokaryote into an indispensable part of the cell, the mitochondrion. This evolutionary leap, the merging of two distinct life forms, gave rise to the eukaryotic cell, the type of cell that makes up all plants, animals, and fungi today. The presence of mitochondria, with their efficient energy production, allowed eukaryotic cells to grow larger and more complex, paving the way for the incredible diversity of life we see today. Mitochondria, often dubbed the powerhouses of the cell, are best known for their role in energy production. Through cellular respiration, they convert nutrients into ATP, the primary energy currency of cells. This process, akin to a cellular power plant, fuels a vast array of cellular activities, from muscle contraction and nerve impulse transmission to the synthesis of new molecules and the maintenance of cellular structures. However, the influence of mitochondria extends far beyond their role as energy generators. They are deeply integrated into a complex web of cellular processes, acting as key players in cell signaling, calcium homeostasis, and even programmed cell death, also known as apoptosis. Mitochondria, with their own DNA, separate from the cell's nucleus, hold the blueprints for a subset of proteins essential for their function. This unique characteristic, a vestige of their bacterial ancestry, underscores their complex evolutionary history. These organelles are highly dynamic, constantly adjusting their shape, size, and number in response to changing cellular demands. They can divide independently of the cell, replicating to meet increased energy requirements or fusing together to repair damage and maintain their integrity. Mitochondria, despite their vital roles, are vulnerable to damage and dysfunction. Mutations in mitochondrial DNA, oxidative stress, and environmental toxins can all disrupt their intricate machinery, impairing their ability to function correctly. This dysfunction can have profound consequences for human health. Because mitochondria are essential for energy production, tissues and organs with high energy demands, such as the brain, heart, and muscles, are particularly susceptible to the effects of mitochondrial dysfunction. The consequences of this dysfunction can manifest in a wide range of health problems, from fatigue and muscle weakness to more serious conditions like neurodegenerative disorders, heart disease, and even accelerated aging. Understanding the intricate link between mitochondrial health and human disease is an active area of research, offering hope for new diagnostic tools and therapeutic interventions. The story of mitochondria is a testament to the power of evolutionary innovation. From their humble beginnings as free-living bacteria to their indispensable role in the cells of all complex life, mitochondria illustrate the interconnectedness of life and the remarkable adaptability of nature. Further exploration of these cellular powerhouses promises to unveil even more secrets about life, evolution, and the delicate balance that sustains our existence.